Okay, guys, please take your seats if possible. The guys on my left, can you please take your seats? Okay, good morning everybody. We are starting here the session of contributed talks on regenerative medicine. My name is Dr. Tzachi Shokovich, I'm an Israeli dermatologist, and with me is Dr. Sofra. There's been a slight change in the chairmans, and since Dr. Sofra is also the first speaker, I'll invite her to the stage to talk about something which for me sounds very interesting: why Nobel-winning studies focus on molecular mechanisms. Please, Dr. Sofra.
Thank you very much for inviting me and for the wonderful presentation. I'm going to talk about why does it Nobel winning prize focus on molecular mechanisms. And the reason why is because the smaller the mechanism, the easier and faster the repair. And also time reversal, going back from part to the whole and back, is routinely adopted for molecular mechanisms. So the physicists have now managed to reverse time at the quantum computer at a very small scale. But what you need to understand is, like, a broken cup cannot go back to being the way it was before. It's always going to remain broken. However, molecular mechanisms like ATP or GTP, they go from AMP to ADP to ATP, and back and forth, and they are exactly whole in all of their formats. And this is basically in accordance with what Einstein said. Einstein said that time, there is a space-time fabric that goes both ways. However, we can only go one way. We can only go forward. We go from past to future. And why is that? Why is it that we can now go back in time? And the reason is because with increasing complexity, as we become a whole person, and basically the parts merge in what we call a gestalt, which is more than the sum of the parts, and it's different than the sum of the parts. So we can all go back in time, because if we scattered ourselves back in time, we would never be the same again. We would never be composed into this whole that is more than the sum of its parts. Also, there is different rules for the old and the young. For example, inflammation is one thing, it's good for the young and deleterious to the old. And there is new theories now. It says there's certain scholastic theories have to do with just repair and regeneration, but there is other theories called semi-programmed theories that they talk about how earlier mechanisms, when they continue, they become counterproductive for us. So the brightest flame casts the darkest shadow. Now, the first Nobel Prize was that I will be talking about is the 2015 in chemistry. And although a broken cup cannot go back to its prior form, however, proteins can routinely repair DNA to make it whole. And DNA repair can happen in two ways: repair and excision. Like we said previously, there certain things have to be subtracted. So thymine dimers inhibit DNA synthesis. So now you have the nucleotide excision of the NER pathway, and basically this is a sequence of how Sankar, since 1983, used different proteins to kind of map this pathway. So we have the basic excision repair, which is called BAIR, and the mismatch repair, which basically you repair the DNA, the part. Again, repair happens at the smaller units and before the entire DNA entity is replicated. So what is the significance of repair in the DNA? Well, DNA creates proteins, and the proteins are basically the alive part of an organism. It's what communicates with the cell, how the cells communicate with each other and with themselves. So basically, if the DNA is damaged, the entire protein production is going to be faulty and it's going to be nonsense, and that all the signaling pathways are going to be compromised. So we know for a very long time, and it's a Nobel Prize in 1999, that the proteins have intrinsic signals that control the entire network of cellular communications. And that's the signal hypothesis. And how they do that is basically they transport signals. And these signals arrive, the way the post has zip codes, they are sort of programmed to arrive to the correct part and offer the signal for optimum production of other proteins or optimum functioning. And this is a protein sequence. Usually it's at the end that signal that is delivered to other cells, just the human insulin and all the letters that are involved there. Now, in Nobel research, that was extended. In 2013, there was a release of neurotransmitters and export of hormones to cell surfaces and communication within cells. And that was like a cargo, like the post delivers packages with food and medicine to different sites of action. Sekman identified a set of genes that are critical for this process. Rothman identified the proteins that are critical for how the proteins bind in specific combinations to deliver the molecular cargo to the right destination. And Shudov identified the molecular machinery that senses, and you see how that protein delivers the cargo via the nutrition and the signals into the appropriate cell structure. Now, the Nobel Prize in Chemistry 2015, now remember in 2015 that they said just that certain mechanisms have to be eliminated for optimal functioning. So you had the base excision and the nucleotide excision. Now, this was extended in 2018. There was another Nobel Prize for eliminating the T cell breaks. And basically, there is two breaks that have been identified. The CTLA4 break, the T cell break, and basically that blocks the T cell to stops it from acting. Now, when you block the break, when you block the TCLA break, then the T cell can attack the tumor cell in cancer and basically kill it. The PD-1 is also another break, and basically, if you inhibit that break, then again you can release the T cells to attack the cancer cells, and there has been a lot of successful treatments of many different cancers, lung cancer, renal cancer, melanoma, and lymphoma with this technology. The activation of T cells, this is how it's presented. It basically requires two things. It requires a binding in a receptor that recognizes a non-cell, and also there is a protein that is an accelerator. Now, these two have to remain as is. However, you have to stop the TCLA break. And when you stop that, then the T cells will attack the cancer cells. PD-1, the exact same thing. Basically, again, when you...
1 minute, please.
Okay. Now, another Nobel Prize had to do with the timing component. That's a very, very important component. And these investigators, they looked at the period gene and the pair protein, and they looked how these two proteins are combined and to kind of produce the effect of molecular circadian clocks. So the importance of molecular circadian clocks is, as you can see here, keloid scars are affected by these clocks, these gene clocks. And also, this disturbance in gene clocks is involved in stretch marks. Also, in psoriasis, you can see before and after, and how when you're adjusting these clocks, how it's been suggested. Now, they're also involved in circadian disruption and cancer risk. circadian disruption accelerates tumor growth, and many other functions. So you have signaling process with respect to time to deliver the necessary cargo with instructions to repair and regeneration. There is another aspect of it. The natural DNA produces nonsense proteins, and this happens during heat, like with radio frequency, for example. Also, the natural proteins deliver this nonsense signals, and the entire network collapses. Now, and there's a lot of disorders as a result of misfolded proteins. Now, the research has gotten a step further. So can we unboil an egg? Can we basically refold unfolded proteins? And the answer is yes, and there is actual a measurement that the negative virial coefficient, this SCV measurements, and -1 indicates that the protein aggregation increased. That means the protein is destroyed, is denatured, and the positive that the protein has a positive result, therefore it decreases, so the protein is going to be more functional. So there is an actual way of measuring this, and this is a video I can show you how this can happen. So corrective signals can amplify faded biological signals, they can fill in gaps in biological signals. As they fill in the gaps, the signals become functional again. And basically, they restore the intra and inter communications between cells, they restore the signaling pathways, and basically communications between cells is equal to health. And with this, I will conclude my lecture. Thank you very much.
Thank you very much, Dr. Sofra. And from the basic science part, we'll move swiftly to Dr. Pace Wam,